Okay, recording is on. Now it's over to you, the organizing secretary, Dr. Shalini Saxena. Thank you.
Thank you, sir. The adventure of life is to learn. And for this, we all have gathered here on a virtual platform for sharing our thoughts. A very good morning to all of you present over here. It's my pleasure to extend a warm welcome to all of you at Invited Lecture Series 4th, organized by Botanical Association, Department of Botany, Bareilly College, Bareilly, in collaboration with Department of Botany, SS College, Shahjahanpur, and SRBS, Society for Research in Biological Sciences. I extend a warm welcome to all of you present over here. First of all, I extend a warm welcome to Dr. Alok Khare, Associate Professor, Department of Botany, Bareilly College, Bareilly, and who is director of the series also. I welcome all the dignitaries and all the faculty members and dear students. I feel extremely honored to welcome today's speaker, Dr. S.K. Bhatnagar, who is former Dean, College of Biotechnology, Sardar Vallabhbhai Patel, University of Agriculture and Technology, Meerut. Sir, we are highly thankful to you. It is such a great privilege to having you with us. Thank you so much, sir. We are really thankful to you for sparing the time out of your busy schedule to interact with our students. I am thankful to all other dignitaries and the dear students. Here is a hearty welcome, big and warm enough to encompass you all. Thank you all. Now I would like to call upon Dr. Asha Singh to introduce today's speaker, Dr. Bhatnagar. Over to you, ma'am.
Thank you, Shalini. Good morning to all. Today we have Professor S.K. Bhatnagar with us. Dr. Bhatnagar started his career in higher education from 1977 as assistant professor. In 2007, he worked as a professor, cell biology, and as dean from 2015 to 17. College of Biotechnology, SVP, University of Agriculture Technology, Meerut. Professor Bhatnagar established internationally recognized Society for Plant Research in the year 1988 under the International Journal of Plant Research, VEGITOS, of which he is editor-in-chief. Dr. Bhatnagar established and started state's first Department of Biotechnology at Bareilly College, Bareilly in 1994. Sir was elected as member of NEC. He is on the expert panel of ASRB, New Delhi. He is the expert of Project Evaluation Council, CCRAS, Ministry of Ayush, Government of India, and many others. Professor Bhatnagar has mentored 16 PhD scholars, a research associate, and a number of MTech students. He has published more than 100 research papers in reputed peer-reviewed journals and many books. He has been honored by the prestigious Vasvik Award 2010 for agriculture research. Sir has a vast experience of 40 years in teaching and research. Besides this, he has worked on several administrative positions. I cordially invite you, sir, to impart your knowledge for us. Thank you, sir. Over to you, sir. Dr. Bhatnagar, sir. Good morning, sir.
Very good morning. Can I have the screen?
सर आप स्क्रीन पे कर्सर ले जाइए सर, तो नीचे प्रेजेंट नाउ आएगा लिख करके.
Present now I have already clicked.
उसमें सर आएगा. आपने सर अपना वो ओपन कर लिया सर, जो प्रेजेंटेशन है उसको ओपन कर लिया सर?
हाँ, yes, it is open.
तो सर प्रेजेंट नाउ में जाएंगे सर तो एंटायर स्क्रीन आएगा. आप जब प्रेजेंट नाउ में जाएंगे तो एंटायर स्क्रीन लिख के आएगा. 3 ऑप्शंस आएंगे, एक ऑप्शन एंटायर स्क्रीन का होगा. आपके लैपटॉप में राइट कॉर्नर पे नीचे आएगा प्रेजेंट नाउ. उस पर जब आप कर्सर ले जाएंगे तो 3 ऑप्शंस आएंगे सर.
इसमें आ रहा है आदर्श जी, शेयर एंड कैंसिल. लेट मी कैंसिल फर्स्ट.
हाँ, तो शेयर करिए सर. शेयर करिए
Share
सर।
now.
Your entire screen. I should
हाँ,
click on
एंटायर स्क्रीन,
right?
एंटायर स्क्रीन सर।
Okay. Then there is share and cancel, but share cannot be clicked.
हाँ सर, तो पहले जो आपको दिखाई दे रहा है सर, जो भी चीज दिख रही है उस पर क्लिक करिए, फिर शेयर ऑन हो जाएगा। जो जो दिख रहा है सर उस पर क्लिक करिए,
Okay, okay.
फिर शेयर जो है वो आ जाएगा। हाँ, इट इज प्रेजेंटिंग सर, इट इज प्रेजेंटिंग।
Now,
लेकिन सर मुझे लगता है हाँ सर, इट्स ओके सर, प्लीज कैरी ऑन। थैंक यू सर, थैंक यू।
is it fine?
Yeah, it is fine, sir. It is
Yes.
fine.
Okay. Very good morning, everybody. Especially my esteemed colleagues, Professor Somesh Yadav, Dr. Ashwini Goyal, and my prestigious student, Dr. Alok Khare, who had been organizing the series so well. I am really obliged for the invitation which has been extended to me by my parent department, where I spent 30 years, and those 30 years are still alive in my memories. Now, when I joined in 1977, it was a small department, but with a perfect coordination, affection, which still continues, even when most of the persons who joined in 1977 or 79 onward, they might have retired or they might have established somewhere else. But still, we are cherishing the old memories. Thank you very much for the entire faculty of the Department of Botany. So I, as asked by Dr. Shalini, to share some experiences and some researches to the students of botany and biotechnology. So the topic I choose is biotechnological tool for agricultural reform, as you can see on the screen. Now let us start from the basic thing why biotechnology and nanotechnology and frontier sciences came into existence. Now everybody, the student of botany and biotechnology or environment science, they might be knowing that for long we were studying various plants, animals, means fauna and flora, their taxonomy, heredity, their anatomical structures, configurations, physiological activities, their metabolism, evolution, so on and so forth. And during our time, there were main biological branches like botany, zoology, chemistry, and so on. So the present day frontier science branches were missing at that time, and we were focused on simply the plant world, the animal world, their biochemistry, their physiology, and all that. With our biological evolution, we multiplied our population many times. As you can see, that the population has gone to 130 crores in India as well, and globally it has reached multiple times. So for constructing our shelter, we started cutting forest, trees, overutilized our water resources, disposed of garbage into water bodies and around. Fertile agricultural land started receding because we started building our residences, we started building towers, and due to urbanization, industrialization, and commercialization, we are having very scarce agricultural land. The conservation of natural resources and food became our top priority beside development. Now, this was the right time when we should start. We started, or the scientists, you can say, started thinking that what should be the way out to combat all these things. Because our population is increasing day by day, our natural resources are receding day by day, our agricultural land is also receding day by day. Then what should we do so that we can cater the entire population? So the scientists started thinking of exploring some frontier sciences. And these frontier sciences were meant to produce more food stuff, more food grains, more opportunities, more jobs, more uh, health or medicinal plants or medicines for the human welfare within a short span of time. Now these branches, as everybody know, now are known as biotechnology, microbiology, biochemistry, food science, nanotechnology, biophysics, environmental science, and so many interdisciplinary branches of science came into existence so as to combat the existing and forthcoming challenges. Basically, what was the need of developing these frontier sciences? Basically, we became instrumental in spoiling our own planet and became prepared for yet another succession by some new races like aliens. Who is responsible for all these things? Why there was a need of frontier sciences? Why there is a need of developing more food crops within 1 year span of time? Why do we want to take more crops in 1 year or in a small span of time? Why do we require more medicine? Why do we require more medicinal plants and natural resources? Because we have spoiled the natural resources which we are having already or which we inherited from nature. Now, different types of technologies came into existence. There is a technology for waste and food preservation. As you can see, this is the scenario, present day scenario you can see. We are disposing of biological or bio and non-biodegradable waste. And these biodegradable or non-biodegradable waste, they are adding to our environment. They are polluting our environment. Maybe the parali burning, maybe the disposal of food waste, maybe the disposal of different types of agriculture waste, or maybe the ritual waste which we had been disposing of in our water bodies. Now the categories of waste you can see in this slide. There are different types of categories of type of waste. I have just given a list of it. Category 1, human and chemical waste, animal waste, microbiology and biotechnological waste, waste sharks, discarded medicine and cytotoxic drugs, solid waste, liquid waste, incineration waste, and so on. So all these type of waste, they are what they are doing. They are adding to our planet, and ultimately, because of these waste, our day-to-day life is being hampered. So we have to find out the alternative sources. As here you can see in the slide, composting of organic waste. The organic waste, the kitchen waste, it may be the agriculture waste, it may be the food waste, it may be the different types of vegetable waste maybe. So these type of wastes are called as organic waste, and these organic waste they can be decomposed by using different types of microbes and can be utilized as a manure or biological manure for cultivating our kitchen garden flowers or plants, or by utilizing it in agriculture fields. Now, this is a slide which is showing the food preservation at Tata Memorial Institute in Mumbai, Atomic Research Institute in Mumbai. I happened to be there for NEC inspection. And during that NEC inspection, I observed that by using different types of radiation, they are preserving different food stuffs. Recently, we had Janmashtami yesterday and day before. So we use kutu, and this kutu floor is not stored for a long time. But these people in biological science department in Atomic Research Institute, by using different types of radiations, they preserved kutu floor and singara floor for a long time, and which was commercialized. So I urged them to make it this technology commercial, so that the people they can get these type of floors throughout the year. Besides these, they were preserving other foodstuffs also for by using radiation techniques and by using different types of radiations. Now there was a need for green energy. Everybody can understand the need of developing technology. Having plenty of petroleum products, we are having plenty of diesel. Then what is the need of it? So everybody should understand that the process of industrialization and with the process of urbanization, we are every time we are requiring these fossil fuel. The fossil fuel reserve is not going to last forever, because this fossil fuel is present under the soil, and it has been developed by regular degradation of different types of biological products for years together. And there is no resource now to produce fossil fuel except consuming it day by day. So for how long we can consume it? For how long this our natural resource can last to cater our requirement of fuel? So we have to find out the alternative sources of energy. Various sources of energy were tried, like wind energy, it was tried, uh, hydroelectric energy we are utilizing, biogas we have also utilized, biomass by using different kinds of tropha and algae we had been utilizing, and we are working on that. That how different types of biomass can be utilized to prepare energy, to form energy, so that our requirement or the requirement of increasing population can be met comfortably. Now, for the student of botany, they can understand that this is a very simple mechanism of converting carbohydrate into uh, energy or into green energy. You can see that all green plants, they can trap solar energy, and algae is one of them. So algae can utilize solar energy. It can synthesize carbohydrates, and along with carbohydrate, it stores fat, lipids, proteins, different types of vitamins. And at one time, algae was supposed to be a very discarded discipline, but for the student of botany and for the upcoming scientists, let me tell that algae and algal biomass is one of the most priority item in the world, because it is capable of producing all type of products. Whatever you dream, whatever you can think, they are capable of producing all these plants. So these algal plants, which are called as microorganisms, they are producing biodiesel, and with the help of biodiesel, the different types of instruments and industries and aeroplanes even can be run. A very simple diagram by which we can use algae for producing green energy in two ways. One, from the industries, which is carbon dioxide and flue gases are coming out. These flue gases contain so many dangerous gases which are harmful to human health and for the plants as well, and for the animal health. So these gases, flue gases, they can be channelized down into the photobioreactor, and this photobioreactor contain algae, and these flue gases, they provide uh, raw material to the algae, so that with the help of this raw material, sunlight and water, they are capable of producing food material, they are capable of increasing their biomass, and oxygen is released. So what happens? The carbon dioxide which is emitted from the industries, that can be converted into oxygen with the help of algae. Second thing, the biomass which this algae has produced, this biomass, what it will do, it will be taken up for transesterification, and by transesterification, its fatty acid will be converted into biodiesel, and this biodiesel can be utilized. The other part, the proteins, vitamins, and other components, they are taken out and they can be used as cattle food, and they can be used for making different types of food stuff. Now, it is very interesting that in 2008, when the National Policy for Petroleum Products came into existence, we organized an international event in Delhi. And you can see that different persons from 38 countries were participating. And in this Algal Biofuel Summit, this gentleman you can see is Mr. Vilas Mutangwa, the then Center Minister for New and Renewable Energy. So we presented different types of models, and in these models, in 2008, the policy by the central government was established that different types of plant products should be utilized to make the petroleum products, and since then, algae and other products were taken into consideration. Now, subsequently, in 2010, we organized a workshop in which the scientists from 8 countries participated. That was also in Delhi, where the people were trained how the algal plants they can be utilized to for transesterification and for the production of biodiesel. Now you can see Professor Michael Borowiska. Now he is the pioneer in making biodiesel from algae, especially, and his student Dr. Navid. Both of them they were the first. They are the first rather to produce biodiesel from algal biomass. And incidentally, in Ireland, I happened to be with Dr. Borowiska, and we had very good discussion about that how India can take lead in producing biodiesel by utilizing algae biomass. Now, another concept in the modern time is that there are various types of algal plants which are capable of producing more fatty acids, which can, through trans-esterification, can be converted into biodiesel. But the point is that can we utilize other algal species also for producing oil, or can we manipulate their genetic setup by using biotechnological techniques so that they become capable of producing fatty acids? There are many algal forms which are underutilized, which are not utilized by the scientists for producing oil. So, if it is possible that we can manipulate their genetic setup and can make them produce oil, so that would be a wonderful thing in the world over. So DNA manipulation to increase the oil producing capacity of algae was one very important target. And in Solar Energy Research Institute, Golden, Colorado, this work has been taken up and it is now almost complete. And the people they are making the genetically modified algal species. Now, this is the setup which we have shown. The US Environmental Protection Agency approved moving things outdoor, and green algal stains were left in water from 5 reservoirs in California. The engineered species known as Acutodesmus dimorphus, and it was designated to synthesize fatty acid and undergo green fluorescent protein expression. So, this is a wonderful outcome of biotechnological research as far as green energy is concerned. Now I come to another very important concept, that biotechnology, what is the role of biotechnology for agriculture reform? Now everybody knows about Dr. M.S. Swaminathan. He is the father of green revolution in India, the first green revolution. The young scientist might be curious to know that why he is called as the father of green revolution. At the time of Lal Bahadur Shastri, we used to import food grain. We used to import wheat from America. Not rather import, but America used to dump the unutilized wheat grains into the sea, and that wheat grains, they were not containing any type of important ingredients or essential ingredients, so they were dumped into the sea downstairs. So America, what he was using, they were using their ships to come to the center of the sea and dump all the food grains. India was starving that time, and they were badly requiring food grains. So Lal Bahadur Shastri was worried about, and that grant, America was told that for halfway you will bear the transport, and for remaining halfway we will bear the transport. And better you do not dump these food grains into the sea, but you dump it into the uh, onto the land of India, so that we can cater our population which is starving. So that grant was called as PL 480, PL 480, and that PL 480 grant came to the heart of Lal Bahadur Shastri, and he started thinking that no more we shall be requiring PL 480 grant, and we will become self-sustainable. So Dr. M.S. Swaminathan was the Director General of ICR, and he was the Director of IARI Usha Institute in New Delhi. So Lal Bahadur Shastri called upon Dr. Swaminathan. He was working with Norman Borlaug, and he was given the responsibility of multiplying the food grain population, and ultimately the green revolution came into existence. Now, the beginning of biotechnology started about 4000 BC in Egyptian discovered a wheat. And then Gregor John Mendel, the student of botany, they very well understand, he is father of genetics. He started finding out that how the characters from one generation move to another generation. Then Watson Crick, he gave the double helical model. Then tissue culture came into existence, where haploid plants and soma clones were multiplied and were cultivated. Then genetic engineering means we started transferring important genes or gene of interest from one plant to another. Then fingerprinting, with the help of DNA fingerprinting, we could identify the biodiversity, we could identify different types of races, and then finally the commercial cultivation of transgenic crop came into existence. We shall discuss a little bit about transgenic crop later in this lecture, because transgenic crop has become a vital issue nowadays, because a lot of debate is going on the transgenic plants. Now, for the beginners, you can simply understand that what happens. We are having a desired gene here, and this is the donor, and this is the commercial variety. We take out the desired gene from here, and we insert this gene into the new variety, and this gene gets associated with the things and finally acclimatized, and a transgenic plant is developed, or a gene with a foreign gene sequence is transferred. Now the development of transgenic plant, you can see that the transgenic plants are confirmed in lab. The transgenic plants are the plants in which the desired gene has been transferred. These are the commercial varieties of the plant. Say, for example, we have developed tomato, and the tomato developed rust, or it is attacked by rust. So, what should we do? We should develop, or we should insert a gene which is resistant for that particular type of rust. So, if we take out this gene of rust resistance from some plant which is rust resistant already, maybe a wild species, and by taking it out, we transfer it into tomato, and then this tomato becomes a transgenic plant, and we have to find out that whether this gene has been acclimatized into tomato or not. So these transgenic plants, they are um, uh, brought to the field, into the greenhouse, and then ecological and biosafety analysis is done, regulatory permit is obtained, then proprietary arrangements are done, and finally the commercial deployment is done, and ultimately the transgenic plants are released. This is the protocol for releasing a transgenic plant variety. Maybe cotton, maybe brinjal, maybe maize, or any other transgenic plant. One of my students has also developed one transgenic tomato. We'll discuss it slightly later. Now there is an apprehension that these transgenic plants, they are very harmful. Now we have given you an example that suppose you are having an iPhone and you are downloading a software into it. It does not become from iPhone to some other phone. It remains the iPhone, but its function is added. So it is just like the transgenic plants are just like adding one character, one gene into a plant, but the plant remains plant. Plant's overall morphology, overall anatomy, and other things of the plant never change. So this is an apprehension that transgenic plants are harmful. So your iPhone is rather much more harmful than the transgenic plants. Now these are two plants, transgenic brinjal and BT brinjal, you can say, and the BT cotton. Then in India, we are not going in details of it, but GM cotton was very beneficial, and the livelihood of farmers in Maharashtra uh, was revived only because of BT cotton. So BT corn is also there. You can see that low mycotoxin, and in BT cotton, it is having a better variety as compared to the normal conventional maize. Now, BT brinjal, which is called as GM eggplant or BT brinjal, it has not been approved by the regulatory body. And my question from different platforms to the regulatory body is that when you grant for a project, and that project is functional, and the people, they start working on that project, and after 3 4 years rigorous research, they are able to release certain variety, then where from this regulatory body immediately uh, jumps up and tells that this is not good for the human health, and this entire research should be kept dumped, or it should be discarded. So this is sheer waste of money from those who are the income taxpayer, who are paying to the government for different researches. So this is very important. Rather, the regulatory body, I appealed with the regulatory bodies and sent a letter to Ministry of Science and Technology collectively that regulatory body should take a decision that what type of research can be beneficial after collecting a data worldwide. Only then the project should be approved for biotechnology. Now this is the value addition. You can see that this is the value addition to different types of crop by using environmental impact. Use of pesticide is there because of biotechnology. Biotechnological interventions, it reduces insects and losses, it increases nutrient uptake and enhances production. GM tobacco can be seen here, and this GM tobacco plant, it helps in cleaning the water toxins. I can give an example to the young researchers that in Meerut, when I was in service over there in university, we were joining hands with Neer Foundation, which is being run by Raman Tyagi and the then Commissioner Mr. Prabhat Kumar. So Kali Nadi is there, which was once upon a time was very good like Ganga, and it was holy and pious. The people used to take its water for drinking purposes of themselves and for animals. Then later on, it became eutrophicated, and the Icornia plant, they completely rather covered the this river, and now this river became Kali Nadi, rather a Nala rather than Nadi. So the plants, GM tobacco plants, were uh, suggested for cleaning Kali Nadi so that the heavy metals or water toxins can be removed. So, through biotechnology, we can also grow different types of plants which are not able to grow in the natural life cycle pattern. Means the natural life cycle pattern, as botanists know, that the plant produce flowers, they produce seed, and seeds are harvested. They are grown again into the soil, and the new plant is developed. Many of the farmers, they used to leave say about 20 or 25% of the plant for seed production, and out of those plants they collected seed, and in the next year they used to cultivate by using those seeds. But there are certain species like human beings, like other animals, they have got the infertility problems. Means the plants are not capable of producing their own variety for one or the other reason. There may be some incompatibility, genetic incompatibility, cytological incompatibility, or any other type of incompatibility. So for those plants, tissue culture has become a boon, and this tissue culture in Elaiocarpus was done, and paper has been published by with one of my students, Dr. Shiva Arshad. She obtained PhD on Elaiocarpus and on Bambusa women, where micropropagation was attempted. Now you can see, as I was talking about the grafted tomato plant. So this was the PhD work of one of my students who worked on tomato, and this tomato is a very important foreign money uh, crops, or you can say a cash crop for India, because much tomato is exported from India to outside. But the problem is that tomato during transit, either it get damaged, or it is infected, or it is damaged during storage or sometimes. So what he did, he took three different plants. One is Nicotiana tovacum or tobacco, which was with the gene of Nicotiana tovacum were introduced to make it disease resistant. Then from strawberry, the genes were taken out to make the skin of tomato thin. And the
Pranam sir,
third was
good
delayed
evening sir.
ripening, which was
Good afternoon sir.
banana. So from banana, the delayed
मैं
ripening
ठीक हूँ ना सर,
genes
आप कैसे?
were taken up, and by help of these three genes, the tomato plants were developed by
हाँ,
the
कब
Batra.
कब आएंगे सर?
So he obtained that the
आ जाएगा
delaying
तो
of
15
cell
अगस्त से
Please
बात कर
keep,
लेंगे.
please keep your mic
16
mute. Please, please, please,
को
please keep your mic mute. Please. Anyway, yeah. Thank you, sir. Please carry on, sir. Please.
So these tomato plants, they were now capable of maintaining their perfect health, even if they are transported to abroad, and its half-life period was reduced, and they were able to survive, say, for about 15 to 20 days later. So it was the senescence was delayed in tomato by one of my students, and it was a very important paper where he worked on these things. You can see different steps for introducing these novel genes. These are the tomato. You can see these are the tomato which are destroyed after 45 days, 10 days, 20 days, 45 days, and these are when RNI was introduced into it. After 45 days, they were as healthy as they were 10 days old. Then again, they were quite healthy even after 45 days. So the shelf life of the tomato was reduced. They were having thin skin, and they were resistant to different types of diseases. This was also a published data with one of my students, Atul Batra. Now you can see, can GM crop solve our problem? This is a very important question, that no single solution is a cure for all. We should not focus more on biotechnology, that only biotechnology can do a miracle and can cure all the things at one time, or you can call it as a panacea. Means we can never do it unless we work in a consortium mode. And this is applicable for everybody. Even if we are doing science, then if we are doing science in consortium mode, then we can bring out something good. But if we are working in isolation, then we can do nothing, and nobody will recognize us. This is the basic fundamental of scientific research. Now, biotechnology can certainly play a significant role. This is a tool which can be used along with different other tools which we are already having, so that we can boost the production of agriculture crops. This is a tool in a toolbox. We are having different types of tools. We cannot say that biodiversity, some people say that biodiversity is lost, but by having biotechnology, biodiversity is also conserved. there are so many red listed species, so many endangered species which are at the verge of extinction. So with the help of tissue culture or with the help of biotechnological tools, we are capable of reviving them. We are capable of reviving multiple this medicinal plant which we are using nowadays, particularly in COVID-19 area, where our immune system is going to be strong with the help of medicinal plants. So we require biotechnology for as a tool in the toolbox. It can fight against world hunger, but it is not only thing which can fight against world hunger. We have to work in a consortium. Can work well with other traditional approaches, as I was just telling about. We must weigh all options, choose the most effective solution, and one should not start blaming one science or one branch of science to another branch of science. Some people say that what is in the basic science. But I have seen the people who are working on biotechnology, who are working on nanotechnology, and they are identifying Ficus religiosa as Solanum nigrum, or they are identifying Solanum nigrum as Eclipta alba. So, what is all that? If we are not knowing the basic science, if we are away from the basic science and we are doing frontier science, then there is no point, because we are always putting absolutely a wrong data before the scientific world, which is more fatal than any other type of research. Now we have just tried to show you food safety, environmental safety, benefits, and transparency. In biotechnological tools, the risks are also there, but the benefits are much more than the risk. So if somebody says that he is at the deathbed and he has to be given this medicine, but this medicine will have some side effect, that what will he opt? He will opt for the medicine with side effect. So this is biotechnology with a little side effect. So can we ignore the benefit and can just uh, just overcome the risk, or we can take some risk and can fight the benefits? This is the slogan that no technology is 100% risk free. Now, why transgenic in agriculture? Imparting resistance to various diseases, insect, pest, and adverse environmental conditions. These are the important issues why transgenics are required in agriculture. Enhancing nutritional value of food, increasing storability of vegetable food. I must tell that very recently I was in US. One of my daughter is residing over there, so I stayed and visited few institutes just for interaction purposes. So I found that most of the foodstuffs, they are much more bigger than we have in India, because they are developing by using transgenic techniques, and the foodstuffs are quite bulky. Maybe their taste may be a little different than the natural crops we are growing in our country, because we are having a lot of agriculture fields as on today, and the most of the people are working and are based on agriculture. But even then, in United States, the people are not doing much agriculture, so they have to import it from outside. So most of the food stuff, they are having enlarged size, and they are having increased nutritional value. Stability of vegetable and food, as I told in case of tomato. Diagnosis of disease is possible. Improving precision and efficiency of plant breeding methods is also there. Transgenics have tremendous potential for the benefit of mankind. So before discarding transgenic or the use of transgenic, like Vandana Shiva, she has been doing, she is the biggest opponent for transgenic plants, and she had been organizing different types of lectures, just not to promote biotechnology, but to show its risks. So I have just told that risks are there. In every technology, there are risks. When Maruti came, there was a risk. When mobile came, there was a risk. When different types of computers came, there was a risk. But we overcome that risk, and ultimately we are now the developed country. So this is the last slide which I wanted to show to my esteemed colleagues and to my students, that this is the Vasvik Award, which is very rare in agriculture sciences and in biotechnology. It carries a cash prize of 1 lakh rupees. And for 2010, it was given by the Director General of CSIR, Dr. R.A. Mashelkar, and it was held in Ramdas Bhaidas Auditorium in Mumbai in 2017. So I am privileged to be a student, to be a teacher of botany and the founder of biotechnology at Bareilly College, and that will always remain in my memory. Thank you all very much, friends, and wish you a very, very good luck. Thank you.
Thank you so much, sir. It was really a wonderful lecture. Thank you so much. It was really a wonderful talk. And Our students learned a lot from your lecture.